girls, where are you going? I'm back. Okay, Nippani class 20 strength looks increased. ಆಗಲೇ ಇದ್ರ ನೀವು ನಿಪ್ಪಾಣಿ ಕ್ಲಾಸ್ 20 you were there? ರಾಯ್ಬಾ ಕ್ಲಾಸ್ 1 ಮಿಕ್ಕಿದ ಮಕ್ಕಳು ಓಡೋದ್ರು ದಾಂಡಿಯಾಗೆ ಅಲ್ವಾ? ಯಾರು ಹೇಳು? ಹೂ ಇಸ್ ಟಾಕಿಂಗ್ ದಿಸ್? ಯು ವಾಂಟ್ ಟು ಗೋ ಟು ದ ಫಂಕ್ಷನ್? ನಾನು ಬರ್ತೀನಲ್ಲ ಮತ್ತೆ. ವಾಟ್ ಫಂಕ್ಷನ್? ವಾಟ್ ಫಂಕ್ಷನ್? Oh, so you all have dressed up pretty. Why Nippani class 20, 19 is not there? Oh, it's a merged. ಮತ್ತೆ ಎಲ್ಲರೂ ಯೂನಿಫಾರ್ಮ್ದಾಗ ಹಾಕಿದ್ದೀರಿ? ಎಲ್ಲಿ ಚೇಂಜ್ ಮಾಡ್ತೀರಾ? ಅಲ್ಲೇ? ವಾ ನೈಸಾ. But you will miss this because Ranebennur is there with us. Divya ma'am class, that is where Dharwad is there with us. So I am going to go ahead with the syllabus. I will not wait. Few question ಮಾಡಿಸಿ ನಿಲ್ಲಿಸ್ತೀನಿ ಅಷ್ಟೇ, ಜಾಸ್ತಿ ಮಾಡಿಸಲ್ಲ. Ranebennur, let's continue. We were hoping that there will be other batches also join, but I don't know, some error. Anyways, so Dharwad, Ranebennur and... No, I am not the person to allow this. I cannot do that. No, I cannot do that. Raiba Keli 1, class 1, Ibrena. Pen ಸಿಕ್ತಪ್ಪ ನಿಂಗೆ? ಕೊಡ್ಲಾ ಪೆನ್? Oh, it was not you. You and he have a similar shirt, I guess, blue and white something. I got confused. Not him, Ray. The other person who was sitting on the same bench. ಮತ್ತು ನೀವೇ ಇಬ್ರು ಯಾಕೆ ಕೂತೀರಿ? ಸುಮ್ಮನೆ ಆ ಸರಿಗೆ ಕಾಡ್ಲಿಕ್ಕೆ ನೀವಿಬ್ರು ಹೋದ್ರೆ ಅವರೇ ಫ್ರೀ ಆಗ್ತಿದ್ರಿಲ್ಲ ಅಲ್ವಾ ಸರ್? ಫಂಕ್ಷನ್ ಸುಮ್ಮನೆ ಏನೋ ಒಂದು ಒಗಿದ್ದಾರೆ. ದೇ ಆರ್ ಇಂಟ್ರೆಸ್ಟ್ ಮೈ ಕ್ಲಾಸ್. ಒಂದರ ಆನ್ಸರ್ ಕೊಡು ಹೇಳ್ತೀನಿ. ಗಿವ್ ದೆಮ್ ಸರ್, ಗಿವ್ ದೆಮ್ ದ ಮೈಕ್, ಗಿವ್ ದೆಮ್ ದ ಮೈಕ್. ಕೊಡು, ಒಂದು ಆನ್ಸರ್ ಕೊಡು. ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಇಸ್ ಮೇಡ್ ಆಫ್ ವಾಟ್? ಅಷ್ಟು ಹೇಳು ಸಾಕು. ಇದು ವಿತಿನ್ ಐ ವಿಲ್ ಬಿ ಹ್ಯಾಪಿ ಟುಡೇ, very happy today. Okay. Nitric. Oh, ito. Pentose sugar, nitric base and some girl was saying something, you know? ರಿಕ್ವೆಸ್ಟ್ ಇಂಟರ್ನ್ ನನಗೇನು ಸಿಗತ್ತೆ? ಯು ಆಲ್ಸೋ ಗೋ ಅರ್ಲಿ ಮ್ಯಾಮ್. ಓಕೆ, ನೋಡೋಣ. ಲೆಟ್ ಮಿ ಸಿ ಹೌ ಮೆನಿ ಕ್ವಶನ್ಸ್ ಆ್ಯಂಡ್ ದೆನ್ ಐಲ್ ಡಿಸೈಡ್. ಅಪ್ಪಿ ರಾಯ್ಬಾಗ್ ಕಾಲೇಜ್ ಕೇಳಿ ಫಾಸ್ಫೇಟ್ ಈಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್, ಸಾರಿ, ನ್ಯೂಕ್ಲಿಯೋಟೈಡ್ ಈಸ್ ಮೇಡ್ ಅಪ್ ಆಫ್, ಇದು ನೈಟ್ರಿಕ್ ಅಲ್ಲ, ನೈಟ್ರೋಜನ್ ಬೇಸ್. NIT ಅಂತ ಶಾರ್ಟ್ಕಟ್ ಬರೆದಿದ್ದೆ ನಾನು. ಇಟ್ಸ್ ಅ ನೈಟ್ರೋಜನ್ ಬೇಸ್, ನೈಟ್ರೋಜನ್ ಕಂಟೈನಿಂಗ್ ಕಂಪೌಂಡ್. ಕ್ಲಾಸ್ 20 ನಿಪ್ಪಾಣಿ, ಆಲ್ ದ ಬೋರ್ಡ್, ಆಲ್ ದ ಐಸ್ ಆನ್ ದ ಬೋರ್ಡ್ಸ್. ಯಾಕೆ ಎಲ್ಲರೂ ಹೊರಗೆ ನೋಡ್ತಾ ಇದ್ದೀರಾ? ಗೋ. ಓಕೆ, ದಿಸ್ ಕ್ವಶನ್ ಡನ್, ದಿಸ್ ಕ್ವಶನ್ ಡನ್. Raise the poll. Raise the card. Dharwad people, you are all there, na? How many nitrogen bases are common in DNA and RNA? Nitrogen bases are component of the monomers of nucleic acid. What is common in DNA and RNA? What three? Which three? Correct? ಹಾ, ನಾನು ಅದೇ ಅಂತಿದ್ದೀನಿ. ಯಾಕೆ ಎಸ್ ಅಂತ ಇದ್ದೀರಾ? And in place of thymine, it is uracilin RNA. So only 3 base pairs are common. Okay, fine. Raise the cue cards. Don't answer, huh? Don't answer. Let me see who can do this.
Ma'am, question is not visible. Yes, ma'am.
Cards. What's your answer, everyone? Wait. Rade Benor is answering C, B. Oh, mixed answers. Raibag, Ibre idira. Can you answer, boys? Try. It's okay. ನೋಡು ನೀನು ಆನ್ಸರ್ ಮಾಡೋದು ತಪ್ಪಿದೆ. ಐ ಕರೆಕ್ಟೆಡ್ ಯು ರೈಟ್. ಬೈಸ್ಕೋತಿ ಬಟ್ ಕರೆಕ್ಟ್ ಮಾಡ್ತೀನಲ್ಲ ನಿಮಗೆ. ಸೊ ಇಟ್ ಈಸ್ ಯೂಸ್ಫುಲ್ ಫಾರ್ ಯು ಓನ್ಲಿ. Okay, the 3 prime 5 prime phosphodiester linkage inside a polynucleotide chain serve to join one DNA strand with other DNA strand, one nucleoside with another nucleoside, one nucleotide with another nucleotide, one nitrogenous base with pentose sugar. Okay, your answer is C. Others?
Option C.
Very correct. Good job. Raibak, good job. What's your name, dear? That's the right answer. One nucleotide to another nucleotide. Iga one base in ಇನ್ನೊಂದು ಶುಗರಿಗೆ ಆಗುವಾಗ what is that linkage? Anybody? Huh? Glycosidic bond. Yes. This bond is called as glycosidic bond or glycosidic linkage. Glycosidic means glucose. Glucose is nothing, glucose or carbohydrate linkage between a sugar and a base, and phosphodiester linkage between a sugar and phosphate group. Here is a ester linkage. And since there are two ester linkage between two, we say it's a phosphodiester. Yes. No explanation needed here, na?
Option A, ma'am. Option A, ma'am.
Wrong. ಯಾಕೆ? Because of this. ಓಕೆ, ನೀವು ಹೇಳ್ತೀರಂತೆ, ಅದಕ್ಕೆ ಅಗ್ರಿ ಮಾಡ್ತೀನಿ. ಓಕೆ, cards, don't say the answer. Hey, ಎಸ್, ಅದು ಹಿಂಗೆ ಹಿಡ್ಕೊಂಡು ಕೊಡಿ. ಬ್ಯಾಸಿಗಾಲಾಗ ಹಿಂಗೆ ಮಾಡ್ಲಿಕ್ಕೆ ಚಲೋ ಆಗ್ತದೆ. ಎಸ್, ನಾಚಿಕಿರೋ ನಿಮಗೆ, ಬಯಸ್ಕೊತಿದ್ದೀರಿ. ರಾಣೆಬೆನ್ನೂರು ಗರ್ಲ್ಸ್, ಸ್ಟಾಪ್ ಇಟ್. Okay, I have answer is D. D. Okay, Dharwad people also D. Okay. Mm, C D. Okay. Nipani class 20? D. Few mixed answers. Okay. Who gave the concept for double stranded DNA ratios between? It's a very clear cut, na? Why A, why B, why C? Anyone has any confusion? There are multiple answers. A, B, C no answer kottidira. Any confusions? No. No?
Option A, ma'am.
Hmm? na? One turn has, yes. ತಡೀರೋ, ಕಾರ್ಡ್ಸ್ ರೇಸ್ ಮಾಡ್ರಿ, ವೇಟ್ ಮಾಡ್ರಿ ನೀವು. ಗರ್ಲ್ಸ್ ವೇಟ್. ಹಾ, ರೈಬಾಕ್ ಬಾಯ್, ಅನಾದರ್ ಬಾಯ್, ಅವನಿಗೆ ಕೊಡು ಮೈಕ್. ಹೇಳ್ತೀಯಾ? ಸರಿ. ಓಕೆ, ಎವ್ರಿ ಒನ್ ಆಪ್ಷನ್ ಸಿ.
Yes, ma'am.
Anti-parallel meaning, anyone? Anti-parallel meaning. Opposite orientation of both the strands. You should say opposite orientation of both the strands is called anti-parallel. ಇದು ಹಿಂಗಿತ್ತಂತಂದ್ರೆ we say it's a parallel, but ಹಂಗಿಲ್ಲ, DNA ಹಿಂಗ್ ಎಕ್ಸಿಸ್ಟೇ ಆಗಲ್ಲ. Okay, what's the answer? Is he right? Okay, be careful. Single strand or both the strand? Why double stranded and why not single stranded? ಅದಕ್ಕೆ ಯು ಕ್ಯಾನ್ ಟೇಕ್ ಅ ಸಿಂಗಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಟರ್ನ್ ಆಲ್ಸೋ. ಇದರಲ್ಲಿ ಎಷ್ಟಿದೆ ಅದು. ಓಕೆ, ಡಿ ಎನ್ ಎ ಕಂಪ್ಲೀಟ್ ಆಗಬೇಕಂದ್ರೆ ಎರಡನೇ ಸ್ಟ್ಯಾಂಡ್ ಬೇಕು, ಹೌದು, ಇದಕ್ಕೆ ಎರಡನೇ ಸ್ಟ್ಯಾಂಡ್ ಬೇಕು, ಗೊತ್ತಾಯ್ತು ನನಗೆ. ಬಟ್ ದೆನ್ ಇಟ್ ಡಸಂಟ್ ಮೀನ್ ದಟ್ ಈಗ ವೆನ್ ಯು ಕ್ಯಾಲ್ಕುಲೇಟ್ ದ ಲೆಂತ್, ಅದನ್ನು ಬೇಸ್ ಪೇರಲ್ಲಿ ಕ್ಯಾಲ್ಕುಲೇಟ್ ಮಾಡ್ತೀರಾ ತಾನೆ. ಹೌ ಡು ಯು ಕ್ಯಾಲ್ಕುಲೇಟ್ ದ ಲೆಂತ್? ಲೆಂತ್ ಆಫ್ ದ ಡಿ ಎನ್ ಎ ಇಸ್ ಕ್ಯಾಲ್ಕುಲೇಟೆಡ್ ಬಿಟ್ವೀನ್ ದ ನಂಬರ್ ಆಫ್ ಬೇಸ್ ಪೇರ್ into distance, correct? So number of base pair ಎಷ್ಟಿತ್ತು haploid content ಅಲ್ಲಿ, diploid ತಗೊಳ್ಳೋಣ because you talk about a whole cell. So 6.6 into ಈ ಥರ ಮಾಡ್ತೀವಿ, which comes approximately 2.2 meters of the length of the DNA, correct? In ಮುಂದೆ we are going to start the same thing. So you can consider single strand also. Then is your answer still same or do you change the answer? Withdraw. I tried to confuse you and you get confused. D only? See, ಅದಕ್ಕೆ ರೀಸನ್ ಜಸ್ಟ್ ಬಿಕಾಸ್ ಡಿ ಎನ್ ಎ ಇಸ್ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಅಂತ ಅಲ್ಲ. ಚಾರ್ಗಾಫ್ ರೂಲ್ ಏನ್ ಹೇಳುತ್ತೆ? ಪ್ಯೂರಿನ್ಸ್ ಇಸ್ ಈಕ್ವಲ್ ಟು ಪಿರಿಮಿಡಿನ್, ರೈಟ್? ಸೊ ಎ ಇಸ್ ಈಕ್ವಲ್ ಟು ಟಿ ಅಂಡ್ ಎ ಅಂಡ್ ಇ ಟಿ ಬರ್ಬೇಕಂದ್ರೆ ಎರಡು ಸ್ಟ್ಯಾಂಡ್ ಬರಬೇಕು ಬೈ ಡಿಫಾಲ್ಟ್. ಯು ಕೆನಾಟ್ ಕನ್ಸಿಡರ್ ಓನ್ಲಿ ಒನ್ ಸ್ಟ್ಯಾಂಡ್, ಕರೆಕ್ಟ್? ದಟ್ಸ್ ದ ಲಾಜಿಕ್. ನಾಟ್ ಬಿಕಾಸ್ ಡಿ ಎನ್ ಎಸ್ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡ್, ದಟ್ಸ್ ವೈ ಯು ಟೇಕ್ ಅ ಡಬಲ್ ಸ್ಟ್ಯಾಂಡ್. ನೋ, the reason behind is adenine is equal to thymine, the purines is equal to pyrimidine. So one strand ಅಲ್ಲಿ ಪ್ಯೂರಿನ್ ಬಂದ್ರೆ ಇನ್ನೊಂದಕ್ಕೆ pyrimidine ಬರುತ್ತೆ, ಅಥವಾ ಈ ಕಡೆ pyrimidine ಬಂದ್ರೆ ಈ ಕಡೆ ಪ್ಯೂರಿನ್ ಬರುತ್ತೆ, and they both are equal numbers. That's why you should consider both the strand. Getting? Okay, let me see if you can do this. Yes. Raise the cards now. Don't anybody say anything. No one will say anything. Okay, we have answers. Great. ಎಷ್ಟು ಎಕ್ಸೈಟ್ಮೆಂಟ್ ಅಲ್ಲ ರಾಡವೇನರ್? ಹಾಂ, ಇನ್ನು ನಾನು ಅಲ್ಲಿ ಬಂದು ಒಂದಿನ ನಿಮ್ಗೆಲ್ಲರಿಗೂ ಬೆಟ್ಟ ಆದರೆ ಹೆಂಗೆ, ಏನು ಮಾಡ್ತೀರಾ? ಹೆಂಗೆ ಕೇಳಿಸ್ಬೇಕು ಅಲ್ಲಿ ಕೂತ್ಕೊಂಡು ಮಾತಾಡಿದ್ರೆ? ಯು ಹ್ಯಾವ್ ಟು ಹ್ಯಾವ್ ಅ ಮೈಕ್. Okay, I have mixed answers. Thank you. I'll also be happy. Mm, ಆಯ್ತು ಹೇಳಿ now tell what Dharwad is answering. Hold on, what Dharwad is answering?
Option B, ma'am. Option B, ma'am.
Mixed answers. Okay, everybody is giving me every random answer. If the total amount of adenine and thymine in a double stranded DNA is 55%, the amount of guanine in this DNA would be. Correct? Amount of ಇದು ಕೇಳಿದ್ದಾರೆ, right?
Yes, ma'am.
A. How? Explain someone.
A plus T divided by G. G plus C must be
A plus T ಏನು ಮಾಡಿದ್ರಿ ನೀವು?
G plus.
ಸೊ 55 ಏನು ಮಾಡಿದ್ರಿ ಮುಂದೆ? ಮುಂದೆ 55 ಹಾಫ್ ಮಾಡ್ಬೇಕಾ? ಏನು ಬರುತ್ತೆ?
100 minus 55.
Cytosine.
Ma'am, G plus C equals to 45.
G plus C equals to 45. ಹಿಂಗೆ ಹೇಳ್ತಾ ಇದ್ದೀರಾ?
Yes, ma'am.
Who is this? Which batch? Which batch? Who is telling this? Class 2? Okay, you are telling A plus T 55 is given, G C is 45. ಹೆಂಗೆ?
Ma'am, G plus C equals to 40. G plus C.
ಹ್ಞೂ ಅದೇಪಾ, ಓಕೆ ಫೈನ್. ಅಂದರೆ what? These two together 45, these two together 55. Correct?
Ma'am.
ಹೆಂಗೆ? How?
100%.
Okay, fine. So assuming that DNA is made up of 100, assuming the total is 100%, ಅದ್ರಾಗಿಂದ 55% is consisting of adenine and thymine. That is what given. Adenine and thymine is made up of making 55%. Amount of guanine, they are asking only guanine. Don't you think ಇದನ್ನು reduce ಆಗಬೇಕು?
Yes, ma'am. Divided by 2. Option D.
This?
Yes, ma'am.
How many agree with this?
Dharwad.
Everybody?
Yes, ma'am.
Anybody who did not understand this, such kind of questions are asked in NEET. Definitely guarantee this. Chargaff rule ಮೇಲೆ ಬಂದೇ ಬರುತ್ತೆ. Anybody who has not understood right now, tell me right now. B. Okay. Oh, my maths is bad. ಹೌದಾ? 45 divided by 2 27 ಬರುತ್ತಾ? ಮ್ಯಾಥ್ಸು ನಾನೇ ಮಾಡ್ಬೇಕಾ? ಅದನ್ನ ನೀವು ಮಾಡ್ರಿ, ಈಗ ನಾನು ಮಾಡಲ್ಲ. See, total amount of adenine plus thymine they have given is 55%. Assuming the DNA is 100%. Huh? D or B? 4 or 2?
4, ma'am.
Huh. So, A plus T ಅಥವಾ adenine thymine pairing 55% ಕೊಟ್ಟಿದ್ದಾರೆ. Then 100 minus 55 is 45. So, guanine ಏನಿರಬೇಕು? Obviously, guanine cytosine 45% ಇರಲೇಬೇಕು. Then only this and this together makes 100% of DNA. But then they are asking the amount of only guanine. ಇದನ್ನ ಬರೇ ಮಾಡ್ಬೇಕಂದ್ರೆ 45ಗೆ ಇಡೀ 45 ಹೇಳಕ್ ಆಗಲ್ಲ. We have to divide it. That is why you get this answer. Understood? Okay. Come on. ಯಾರದು? ಕ್ಲಾಸ್ 2. ಐ ಲವ್ ಯುವರ್ ವಾಯ್ಸ್ ಗರ್ಲ್. ನಿನ್ನ ಹೆಸರು ಹೇಳಪ್ಪಿ. ಹೇಳೋ. ಆ ಏನು ಆಪ್ಷನ್ ಅಷ್ಟೇ ಕೇಳಿಸ್ತು. ವಾಟ್ ಆಪ್ಷನ್?
Option C.
ಸಿ.
C1.
Incorrect. You have to find incorrect.
Yes, ma'am. Is the incorrect answer. C option.
I want her name. What's her name? Ranebendur class 2. I want her name. ನೀವೇ ಅವಳು ಹೇಳಲ್ಲ, ನೀನೇ ಹೇಳೋ. Class 2 middle bench, ಹಾ. The one who is giving like this mic. ಆಟ ಆಡ್ತಿರಲ್ಲ ಮೈಕ್ ಜೊತೆ. Not you, not you, not you. Ranebendur class 2 I'm talking. Ha. What? Meghna? Hi Meghna, you look very pretty. What's her name? Who is Asha? Okay, next to you? Ha. So it is very nice that you are very interactive. And Meghna, it's the only you who keep on telling me answers every time? Because ninde dhani kelutte nange. Okay, good. Nice interacting to you. Fine Meghna, your answer is also C. Okay, ನೋಡೋಣ. Incorrect ಹೇಳ್ತಾ ಇದ್ದಾರೆ. ಚಾರ್ಗ್ರಾಫ್ ಆಟೋ ರೇಡಿಯೋಗ್ರಫಿ ಆಫ್ ಹೌದಲ್ಲ, ಕರೆಕ್ಟ್. ಬಾಕಿ ಮಿಕ್ಕಿದೆಲ್ಲ ಕರೆಕ್ಟ್ ಇದೆ. ಎಫ್ ಮಿಶ್ಚರ್ ಡಿಸ್ಕವರ್ಡ್ ನ್ಯೂಕ್ಲಿಕ್ ಆಸಿಡ್ ಅಂಡ್ ನೇಮ್ ಇಸ್ ನ್ಯೂಕ್ಲೀನ್. ವಿಲ್ಕಿನ್ಸ್ ಅಂಡ್ ಫ್ರಾಂಕ್ಲಿನ್ ಎಕ್ಸ್ರೇ ಡಿಫ್ರಾಕ್ಷನ್. ವ್ಯಾಟ್ಸನ್ ಅಂಡ್ ಕ್ರಿಕ್, ಎಸ್. Okay, fine. What is central dogma of a cell? What do you understand by the meaning central dogma of a cell? Slow and louder. Meghna it is? Allah, this is not Meghna. Who is this? Asha? Tanushree. Which class? Class 1. Ha, genetic information that flows from? Correct? And so, the whole DNA, athwa, the whole process of the cells in your metabolism completely depends upon this central dogma. DNA is going to undergo the process called replication, where it makes its own copy. This is simply making the copy of its own. Generally, this happens only when cell undergoes cell division. S phase. You remember S phase? PUII, cell division, mitosis, meiosis, interphase. There, G1 phase, S phase, G2 phase ittu. S phase alli content of DNA doubles, that is replication. This is what they were talking about. So, remember, replication or copying of DNA is not a constant process or not a continuous process. DNA will not keep on making and copying the DNA continuously. It would only make a copy of DNA when the cell has to divide. Illantandre madalla. Simply it will make the other two processes, that is transcription and translation. So process of conversion of DNA to RNA is termed as transcription. Means we are simply changing the language of DNA to RNA. That is nothing but changing the thymine nucleotide to uracil. And then when this RNA, we have changing the language of RNA to amino acids. Proteins are made up of amino acids, and amino acid chain is called as polypeptide or protein, simply. And lastly, out of the nucleus, out of the cell, when this protein comes, this is used for various functions of the cell. Protein can go and form an enzyme, it can go and form a receptor, it can go and form a hormone, any other molecule wherever required. So DNA is, that's why called the source or a master or the brain of a cell, because adrinda baro yen protein ide, that is involved in the actual metabolism. Okay? Now, the packaging of DNA, just now we calculated the length of DNA, you know. So the length of DNA, when the human DNA is almost 2 meters, how do you think this would fit in such a small cell? Or 2 meters is something this big. the board is around 2 meter, ಇದಕ್ಕಿಂತನೂ ಸ್ವಲ್ಪ ಜಾಸ್ತಿ ಆಗ್ಬೋದು, but this big. So how coiling, yes, correct. But give me one idea, people, when I say about 2 meters of the DNA, roughly 2 meters of the DNA, do you understand that 2 meters of the DNA is of only one cell? Or do you understand that 2 meters of the DNA is of the whole content of the DNA from your whole body? What do you understand? You are made up of billions and trillions of cells. So, ಎಲ್ಲ cells ಸೇರಿ DNA ದು one length 2.2 meters ಆಗತ್ತಾ? Or one cell ಅಲ್ಲಿ 2.2 meter DNA ಇದೆ? What? Correct. One cell. So, when one cell is so tiny that you cannot even see with your naked eyes, how do you think they are going to, in case of E. coli only, what they do? Generally, there are some protein, E. coli athwa you can say prokaryotes. There are some proteins that will help the nucleus or the nuclein, the DNA, to come in a compact region. Prokaryotes do not have nuclear membrane, right? Prokaryotes do not have nuclear membrane. Gottide alva?
Yes, ma'am.
So does that mean that in a cell, oh my god, what a beautiful cell. Does that mean that in a cell, this DNA has to be scattered everywhere? No. DNA is generally placed only at one point in loops, even though there is no nuclear membrane. But on the other hand, if you take a eukaryotic cell, there you have a nucleus inside which the DNA would be properly placed in a loops again. So in both the cases, there is something that binds the DNA. This binding of the DNA is because of the proteins, some positively charged proteins. Why? Why positively charged protein?
Due to presence of the amino acids.
Okay, protein is positively charged because of the amino acids, gottaito. But a positive charged protein ಯಾಕೆ ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದೇವೆ? Why the DNA is, means protein plus DNA is together called as chromatin. Protein, yes, who is it? Why DNA is negatively charged? Basic, people, this will be very fun. You will love learning this. Why DNA is negatively charged? Dharwad, speak. Why
Presence of phosphate group.
Exactly, it is a phosphoric acid giving UPO4 minus, nucleic acid, negatively charged. So because the DNA is negatively charged, we use some positively charged proteins. Name of the proteins in prokaryotes is not given, but yes, definitely in eukaryotes we know. What is that proteins?
Histone protein.
Yes, that histone proteins that is present in octamer. What is octamer? DNA is wrapped around the histone proteins anta ಹೇಳ್ತೀವಿ. The DNA will be wrapped around the histone proteins like this. But histone proteins are octamer. Octamer means which eight? Which eight? Which 8 molecules? Nobody knows? These 4 histone molecules are twice in number, and that's how that becomes a octamer. H2A, H2B, H3 and H4, all this together are 2 times in number. 2H2A, 2H2B, 2H3 and 2H4. So I have written H2 into 2 anta. So that's how they become a octamer, or that's how they become a one group of octamer called as histone proteins. Along with that, it has one more protein called as H1, which is called linker molecule. I will show you this. This is visible? So you can see 1, 2, 3, 4, 5, 6, 7, 8. This together whole ball of 8 molecule is called as octamer, histone proteins. And why histone proteins? As she said, because histones are positively charged. They are rich in lysine and arginine. These are positively charged amino acid. Hangantheli bere amino acid illa anta alla. Again, they will have different amino acids also, negatively charged also, but the amount of the positively charged amino acids is more compared to others. That's why we say that histone is positively charged. They are rich in lysine and arginine. So this 8 molecule forms an octamer, and it wraps almost how much of DNA? No, 200 base pairs. One nucleosome. This is called what? This is called one nucleosome. 200 base pairs of DNA plus one octamer set is called as one nucleosome. And like that, there are many nucleosomes. Like this. So this one wrapping is 200 base pair, 200 base pair, 200 base pair, 200 base pair, and these two, this is one nucleosome. This is one nucleosome. This is second, third, fourth. Nucleosome to nucleosome is one connected by H1, which is called linker. This, you can see this. Understanding? Are you able to understand the concept? Nucleosome model clear? Everyone? Okay, what is the next thing in your book now? Nucleosome ಆದ್ಮೇಲೆ ಏನಿದೆ? Search, ಬೇಡ, ಸರ್ಚ್ಗೆ ಈಗೇನು ಸದ್ಯಕ್ಕೆ ನಾವು ಸರ್ಚ್ ಮಾಡಂಗಿಲ್ಲ. We'll do only till nucleosome. Am I missing anything? Chromatin ಹೇಳಿದ್ನಲ್ಲ. Heterochromatin, euchromatin, yes. Chromatin is nothing but DNA plus proteins are together called as chromatin. People, one more, so you may come across a question, histone word ಏನಿದೆ, histone word of the protein is only applicable to eukaryotes. Prokaryotes ಅಲ್ಲಿ histone proteins ಬರಲ್ಲ, but histone like proteins, similar proteins ಬರುತ್ತೆ, not histone. And secondly, there should be NHC complex also. Non-histone like chromosomal proteins anta irbeku, underline somewhere. Non-histone like chromosomal proteins, NHC complex. So bari histone anta alla, along with histone there are non-histone proteins also that help the DNA to compact. Idella yak ಮಾಡ್ತಾ ಇದೀವಿ ಕಥೆ? Why are we doing this? Because we want to compact the structure of DNA into the one small tiny nucleus. By doing this, what will happen? See, this appearance is called beads on a string appearance. Beads muttin sarathara, simply. You have a thread, then you have a bead. You have a thread, then you have a bead. You have a thread, you have a bead. So, idina, what we are doing, we are trying to coil, we are trying to compact. Mundinagatte, this structure is called as now chromatin, okay, where there is a protein also, where there is a DNA also. Now what happens? This structure, when it starts compacting over each other like this, like this, like this, like this, will condense and get more condensed and forms the structure something like this. Then we say it's a chromosome. Idane DNA part, this part of a chromosome, if you unwind, ಸ್ವಲ್ಪ ಝೂಮ್ ಮಾಡಿ ನೋಡಿದ್ರೆ ಇದನ್ನೇ ಈ ಥರ ಕಾಣುತ್ತೆ. ಇದನ್ನೇ ಮತ್ತೆ ತಿರ್ಗಾ ಝೂಮ್ ಮಾಡಿ ನೋಡಿದ್ರೆ it will look like this, and if you zoom it will look like this. So chromatin is a structure of a DNA where it is present only proteins and DNA molecule. That chromatin, when it coils and condenses, becomes a compact structure. Then we call it as a chromosome. Again, this structure is taken place only when the cell has to undergo division, prophase, metaphase, anaphase. Only at that time the cell will take this structure. Why? It is simple logic. ಇಷ್ಟುದ್ದ ನಾನು ದಾರ ತಗೊಂಡಿದ್ದೀನಿ, ಓಕೆ. ಇಷ್ಟುದ್ದ ದಾರನ ಸ್ಮಾಲ್ ಬಿಟ್ಸ್ ಅಲ್ಲಿ ಕಟ್ ಮಾಡ್ಬೇಕಂದ್ರೆ ಐ ಹ್ಯಾವ್ ಟು ಯೂಸ್ ಸೀಸರ್ ಸೋ ಮೆನಿ ಟೈಮ್ಸ್. ಸಿಂಪ್ಲಿ ಆ ಅಷ್ಟುದ್ದ ದಾರನ ವಾಟ್ ಐಲ್ ಡೂ, ಕಾಂಪ್ಯಾಕ್ಟ್ ಮಾಡಿ ಇಷ್ಟೇ ಮಾಡಿ ಒಂದೇ ಕಟ್ ಮಾಡಿಬಿಡ್ತೀನಿ. ಈಸಿ? ದಟ್ಸ್ ಮೋರ್ ಈಸಿ ನಾ? Simple. So cell division is what? You want to divide the number of chromosomes equally. ಈಗ 2 meter DNA ನ ಹೆಂಗ್ equal divide ಮಾಡ್ತೀರಾ? Practically not possible. And everything is happening in a small tiny cell. So DNA is also smart. It also does the job of compacting everything and taking a structure of chromosome. We generally draw chromosome pair ಈ ಥರ ತೆಗಿತೀವಿ ನಾವು, okay. ಅದು under the microscope it was looking like this, that's why we draw it like this, but it is not literally like this. ಸ್ವಲ್ಪ ಉದ್ದುದ್ದ chromatids ಇರುತ್ತೆ. And chromosomes are never separate from each other. There is a DNA which keeps on holding them. Aden separate ಅಲ್ಲ. It doesn't mean that DNA is cut into small pieces. No, but that DNA is more tiny that under the microscope you cannot visualize it. ಕಾಣಲ್ಲ. So we assume that it looks like this, but we should be having in your mind that hangalla, they are connected. Okay, so that was about your chromosome, chromatin, and euchromatin ಉಳಿದಿದೆಯಲ್ಲ. Okay, so the when the DNA is tightly packed at some places and it is dark stained, then we say it is what? Heterochromatin. And heterochromatin is transcriptionally inactive. Inactive ಅಂತಂದ್ರೆ ಏನು? ಇಷ್ಟು ಉದ್ದ DNA, this is a big DNA of 2 meters. This DNA only, I have taken the shape of 23 pairs of chromosome. In what happens, certain places of DNA there is a repetitive sequence. DNA is highly repetitive. 98% of DNA has repeating sequence of base pairs. Only few places of DNA has a factor called as gene. That was what Mendel was trying to say. So suppose the gene is only at somewhere here. Rest of the parts is having simply the repetitive paths. Central portion, generally the central portion. This is not going to code or be inactive transcriptionally, means it will not give you any mRNA, it will not give you any gene, or it will not give you any protein. Whereas DNA, where it is little bit loose bound, that is called euchromatin, and this is transcriptionally active, which means this is the part of a DNA from where you're going to get a protein. Okay? Now also you still feel this was complicated or salpa relax aitu? You want me to tell somewhere again? Watch ಏನು ನೋಡ್ತೀಯ? ನನ್ನ ಹತ್ರನೂ ವಾಚ್ ಇದೆ. Those who felt the chapter is complicated, at least we have started with the basic. Is it clear? Anything, then ask now. See, ಈಗ ಕಾಲೇಜಲ್ಲಿ ಒಂದ್ಸತಿ ಆಗಿದೆ, ಇಲ್ಲಿಯೂ ಒಂದ್ಸತಿ ಆಗಿದೆ. In ಮುಂದೆ when you give the test again, if you fail, then it will be like you are not understanding at all anything. So when you have a second opportunity, ask it. ಸೆಂಟ್ರಲ್ ಡೋಗ್ಮಾ ನಿಮಗೆ ಗೊತ್ತಿದೆ. ಐ ಆಮ್ ಸ್ಕಿಪ್ಪಿಂಗ್ ದಿಸ್. ಡೂ ದಿಸ್. ಅದನ್ನು ನೀವು ಮಾಡ್ಕೊಳ್ಳಿ ಆಮೇಲೆ PDF ಅಲ್ಲ. ಸಿ ಅಲ್ಲ, ಬಿ 200 ಬೇಸ್ ಪೇರ್ಸ್ ಆಫ್ DNA. ಓಕೆ. ಅರೆ Okay, now wait for the next question. Raise the cards. See, They have seen so beautifully. This is called one nucleosome. This is called one nucleosome. And many nucleosome and DNA come together. This is called what? नहीं, chromatin. Pa, ಇಲ್ಲಿ ನೋಡಿ, this histone protein is one octamer. ಓಕೆ, ಆ ಒನ್ ಆಕ್ಟಾಮರ್ ಸುತ್ತ ಇದೇನು ಡಿ ಎನ್ ಎ ಇದೆ ಅಲ್ಲ, ದಟ್ ಈಸ್ ಆಲ್ಮೋಸ್ಟ್ 200 ಬೇಸ್ ಪೇರ್ ಡಿ ಎನ್ ಎ. ಈಗ ಇವು ಎರಡು ಆಕ್ಟಾಮರ್ ಲಿಂಕ್ ಆಗುತ್ತೆ ಬೈ H1 ಲಿಂಕರ್. ದೆನ್ ಆಕ್ಟಾಮರ್ ಆ್ಯಂಡ್ ದ ಡಿ ಎನ್ ಎ ಟುಗೆದರ್ ಇಸ್ ಒನ್ ನ್ಯೂಕ್ಲಿಯೋಸೋಮ್ ಮಾಡೆಲ್ ಆರ್ ಒನ್ ಸಾಲಿನಾಯ್ಡ್. ಇದಕ್ಕೆ ಸಾಲಿನಾಯ್ಡ್ ಮಾಡೆಲ್ ಅಂತಾರೆ. ಈ ಥರ one nucleosome, 2, 3, 4, many, all there in a chain, beads on a string appearance called as chromatin. Okay. And now no answering, raise the card. Okay. A histone, C DNA, B H1 histone, C histone octamer, A H1 histone, B DNA, A histone octamer, B H1 histone, C histone octamer. Howdo? Yes. Yes, most of you are right. Okay. Raise the cards, Ray. Hey, can you see this diagram and observe, people? Ill nodi, this area is so tightly packed called as heterochromatin, whereas this area is a little bit loosely packed called as euchromatin. And ಅದರ ಮೇಲೆ RNA polymerase ಬಂದು ಎಷ್ಟು ಚೆನ್ನಾಗಿ ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಮಾಡ್ತಾ ಇದೆ. RNA polymerase transcription. So that's why we say euchromatin means which is a loosely packed, that is the area where actually protein synthesis happens. Tightly packed area ಅಲ್ಲಿ protein synthesis ಆಗಲ್ಲ, ಅಲ್ಲಿ ಬರೀ ರಿಪಿಟೇಟಿವ್ ಡಿ ಎನ್ ಎ ಇದೆ, ಅಲ್ಲಿ ಜೀನೇ ಇಲ್ಲ. ದಿಸ್ ಇಸ್ ಯುವರ್ ಜೀನ್. ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಜೀನದಾಗತ್ತೆ. ಓಕೆ, ಟ್ರಾನ್ಸ್ಕ್ರಿಪ್ಷನ್ ಹ್ಯಾಪನ್ಸ್ ಆಫ್ ಜೀನ್. ಹಾ, ಏನು ಹೇಳಿದ್ರಿ ನೀವು? ನಾಟ್ ಅ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್, ನಾಟ್ ಆಫ್ ಹೆಟ್ರೋಕ್ರೋಮ್ಯಾಟಿನ್. Okay. ಇವ್ರು ವೇಟ್, A3B. Okay. ಹೌದು, euchromatin is loosely heterochromatin. Okay, yes, yes, yes. Fine. ಇದು ಈಸಿ ಇದೆ, ಬೇಡ. ದಿಸ್ ಇಸ್ ಆಲ್ಸೋ ಈಸಿ, ನೀವೇ ಮಾಡ್ರಿ ಇದನ್ನ. ಬೇಡ, ನೋ ನೋ ನೋ. ಡೂ ದಿಸ್. ಐ ವಾಂಟ್ ಸಮ್ ಟಫ್ ಕ್ವಶನ್. ಡೂ ದಿಸ್. ಹಾಂ, select the correct option. Easy, 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 easy. Genetic. Many of you are going with A. Ranebenur A. Repani also A. Okay. Genetic material has a validity of self-duplication. Genetic material does not express itself in the form of Mendelian character. Does not? Tappo. Genetic material is very stable and does not change. No. Mutations agatte tane. We have seen mutations. Genetic material store information to control cellular function. True ala? Genetic material store in, but not all genetic material, only genes store. This should be genes to control the cellular function. Okay, yes, which is the correct option they are saying. Okay. On their question, purines and pyrimidines ಬಗ್ಗೆ ಇದೆ ಈ PDF ಅಲ್ಲಿ, you can just solve it on your own. So we will resume with the search of a material, Griffith experiment. It's interesting, people, don't miss the next class. Friday and Saturday, next week Friday and Saturday you will have similar classes like today, 1 hour classes irutte. And all the best for your Saturday's test. Good luck. It is a Mendel's chapter, next half of the chapter, I hope you remember. And finally, have a happy Dasra, enjoy your festival, good day. Somebody saying something? Huh? I will not be able to tell you. You can ask the teachers. Nange ಹೇಳಕ್ಕಾಗಲ್ಲ. You will be getting it. Message ಬರುತ್ತೆ. Okay. Anything else? Syllabus is clear?
No, ma'am.
Okay. Have a good day. Enjoy the function. Bye bye. Feedback. Generate the feedback card, people. Bye bye. Show the feedback card quickly. Teachers, please do the needful. ನಾನು ಒಂದು ಫೀಡ್ಬ್ಯಾಕ್ ತೋರಿಸ್ಬೇಕು, ನಾನು ಇನ್ಮೇಲೆ ನಿನ್ನ ಕ್ಯೂ ಕಾರ್ಡ್ಸ್ ತೋರಿಸ್ತೀನಿ ಅಲ್ವಾ. ಯು ಶುಡ್ ಆಲ್ಸೋ ಗೆಟ್